Everyone, it's my pleasure to introduce our speaker for today, Dr. Doug Zochodne. So he hails from the University of Calgary. He did his MD and his neurology residency at the University of Western Ontario. Then from there he went on to the University of Calgary, where he climbed the ranks as assistant professor to full professor. He's also wears many hats, so he's the director of the clinical neurophysiology and EMG unit, Calgary Health Region. He's also the director of the Neuromuscular clinic and the staff neurologist at UofL Hospital. And he's had numerous awards and distinctions, and he's published over 240 papers to date. He's a world expert in peripheral nerve regeneration and also different modeling in his lab, diabetes and different neuropathies as well. So Doug's title today is New Axons, the Challenges and Beauty of Peripheral Nerve Regeneration. You're welcome, Doug.
Thank you very much, David. It's a real pleasure to be here. I think I'm getting the name right in my head, Louis Ball. Maybe still not quite right. It's a real delight. It's a very nice center that I've been privileged to be invited to, with some really exquisite work that I just started to learn about yesterday. You know, beautiful microvascular work by Dr. Benton and electrophysiology with Dr. Magnuson. And some really interesting work with children with Dr. Bierman yesterday. So fascinating, and I think you're very fortunate to acquire Dr. David Sterling, who I've had really high regard for in Calgary, and I'm sorry we've lost him, but he's an outstanding neuroscientist. So beautiful spinal cord work being done. My talk's a little different, but we have a lot of commonalities between the spinal cord and peripheral nerve, and we hope some of the targets, and we know some of the targets, will be shared between these two systems. So I'm going to talk a little bit about peripheral nerve regrowth, emphasizing just hitting on some new molecular pathways that we're excited about and interested in, and just covering some of the background of it a little bit. I guess the first argument I usually try to make is that peripheral nerves are unique. They're a distinct tissue. They have distinct components, the epineurium, endoneurium, where the axons are, either large myelinated or small unmyelinated. Every part of the peripheral nervous system is subjected to different types of disorder, and they're very different disorders than spinal cord injury, CNS degenerative diseases. They're very unique and interesting. They're under the radar often as well. It's a mouthful, peripheral neuropathy. But our awareness is rising slowly. In fact, in mid-May, if you're near Niagara Falls, you may see the lights on the falls change color for Neuropathy Awareness Week. So we can accomplish a few things like that. Have we solved peripheral nerve disorders? No. Okay, so some of the unique aspects of neuropathy they're incredibly common. Diabetes is burgeoning worldwide. Some recent statistics saying that it may outpace even dementia by tenfold. And polyneuropathy, or diffuse disorder of the peripheral nerves, is present in up to about 50% of diabetics if you test it carefully. Symptomatic, maybe 10 or 20%. So very, very common neurodegenerative problem that targets the longest nerves and causes such a pattern called stalking and blood sensory loss. It's an intractable disorder. There's no way to arrest or reverse this disorder right now except perhaps having very tight control of hyperglycemia, even that not successful. So these are acquired neuropathies for which there are huge challenges. And even if we were to arrest this, the islet transplantation, for example, to get the nerves to grow back to their targets where they belong is the major challenge. But there are many other types of peripheral nerve disorders. This is a woman with brachial plexopathy from radiation. She had breast carcinoma, and she has a wasted weak hand and may have neuropathic pain as a result of this. 100 years has passed since Tennell produced this slide of somebody with a musket injury to their sciatic nerve, Even in this day and age, in 2014, with the best microsurgical neurosurgeons and nerve surgeons reopposing this nerve, to get it to grow back below the knee for function below the knee is non-existent right now. So many challenges in terms of the regrowth and repair of the peripheral nervous system, but a little bit under the radar. You don't hear about it as much as things like dementia and cardiac disease, for example. So lots of techniques surgically around, and there's no doubt that these have helped during gap injuries, etc., but not the answer. The neurobiology of the peripheral nerve is also very unique and very interesting. We do bridle a little bit when people say, oh, well, that's a great model of the central nervous system, where it really counts, where it's really important. The further these nerves have to regrow, and the longer the delay between the injury and the time of regrowth, the less likely they are to reinnervate their targets. So the disabilities with peripheral nerve disease are huge. And we see them in clinic, whether it's in a diabetic or someone with HIV, small fiber neuropathy, very, very common. Having said that, I think there'll be a lot of commonalities, as I mentioned earlier, between central white matter disease and the peripheral nervous system. But the facets of the neurobiology are interesting, and there are there's a smaller cohort of investigators that look at these problems, but have addressed specific aspects of this. So, for example, if you transect this pseudo-unipolar sensory neuron area, you have some central changes in the cell body that occur. The nucleus migrates peripherally. These perineuronal satellite cells proliferate and enlarge. And there's a whole series of changes in the cell body that are reprogrammed that I won't be talking a lot about today. And these are called RAGs, or Regeneration-Associated Genes. One of my colleagues, Valerie Verge at the University of Saskatchewan, has been a major investigator of RAGs and how they occur and when they come on, and artificially inducing RAG expression with things like electrical stimulation. So there's many other steps in this sequence. So there has to be sprouting from the proximal stump. Macrophages invade this degenerating nerve. The process of axonal degeneration has turned out to be quite interesting. So this has also been termed Wallerian degeneration, which strictly speaking refers to a knife edge injury and a distal series of changes in the nerve. We used to think this was a passive process. The cell body's cut off and these guys die. But people more recently had recognized that it's an active molecular program, axonal degeneration. There are mice in which the axons persist for long periods of time, WLDS mice. And so that whole story is proving to be very, very interesting. Why would you want to preserve this? Well, it's not a great idea to preserve it if you want the axons to regrow. So the success of subsequent axon regrowth depends on how this debris, so to speak, is cleared by Schwann cells here and macrophages. And there are inhibitory components, just like in the spinal cord, to prevent the regrowth. So there is myelin-associated glycoprotein and other constituents. But there are neuropathies in which damage to the axon is primary. And so we see those often in chemotherapy-induced neuropathies, where there's a primary axonopathy. And in those instances, it would be absolutely superb to be able to protect these axons against these agents that are essential to shrink tumors. Okay, so I mentioned sprouting, which is something we've been particularly interested in. The success of the inflammatory response, and this would be analogous to work in the spinal cord that David would be working on. The importance of a sterile inflammatory response in terms of clearing debris and allowing sprouting to occur. The proliferation of the Schwann cells here to guide the axons. You may not want too many sprouts here because they may compete for growth factors and make regrowth of a successful branch more difficult. So then you have a maturation of the axon, maturation of the nodal apparatus. That's only part of the story, because out here somewhere you have your target tissue, and so even though the axons mature, for it to reach all the way into the skin, into the epidermis, periosteum, etc., is another story. And I think David was telling me yesterday that this center is a pioneer in hand transplantations, and some of that literature is really very interesting because even if you sew together 3 of the nerves, when you transplant the hand, and then you do epidermal biopsies, this was work done in Sweden, to look at epidermal fiber regrowth, it's really restrained and limited even 5 years down the road. So I think technically the surgeons have been very adept at doing amazing things in terms of reconnecting these hands. The trick will be to get them reinnervated normally, the muscles and the sensory organs. So here's a cartoon of some of these events: motor and sensory neurons, the injury, axonal degeneration, influx of macrophages, proliferation of the Schwann cells, degeneration of the axons. These parent axons form little end bulbs that are interesting, and they release vasoactive substances into this milieu that may dilate vessels. The axons may not necessarily know which way to grow, so directionality, we think, is important. They may find Schwann cells and have a roadmap to travel back to where they belong, but initially they may grow wrong ways. Okay, so I've touched on a couple of things here. The pace of this axonal degeneration or Wallerian degeneration, Wallerian-like degeneration, does influence subsequent regenerative success. So if you poison macrophages, or as we did, if you have a mouse with a knockout of nitric oxide synthase, an inflammatory mediator, the Wallerian degeneration is delayed, and then subsequent regeneration is delayed. The Schwann cell is an essential player and partner for guiding growth. So we think in the vast majority of instances, the Schwann cell leads out, migrates, but the axon follows very, very closely. And the Schwann cell changes its phenotype. It becomes activated, becomes elongated, and there's this very close interaction between the axon and Schwann cell. We've called this the axon-Schwann cell dance. We try not to stretch the analogy too far, but there is an interchange between the two of them of molecules back and forth to help one another, and I'll touch upon that. So David McDonald was a PhD student in the lab. He's gone on to be a plastic surgeon in, like a number of people in our lab, in Red Deer, which is about 150 kilometers north of Calgary. And we set up these chambers with David. Now, chambers like this, reconnecting nerves, have been around for a while. Lundborg in Sweden pioneered them with silicone, and my colleagues in Calgary are exploring biodegradable chambers, etc., to reconnect nerves. But the reason we set this up was to be able to influence this regenerative milieu and look at what it may take to rebuild a nerve bridge here, to have the axons grow from the proximal to the distal stump, interact with the Schwann cells, and regrow. And it's quite interesting that a completely new nerve can form in these gaps. So many of you know that when you transect a nerve, the proximal and distal ends retract a little bit. And that's probably because of some elastin and they're under some tension. So there are these gaps, and I think neurosurgeons can, I'm not a neurosurgeon, but they can bring them together in some instances with suture. But sometimes it's impossible or there's too much stretch involved. So you need to have a tube like this. But gaps are a big part of the challenge of peripheral neurobiology. Tom Brushard at Hopkins and Witzel estimated that if there's a gap, and even if you do oppose the proximal and distal stump, just the transection alone, there's only a 1 in 10 chance of the proximal axon reaching its target and reinnervating. So a huge challenge to regrowth. Right, so one of the things that we've been interested in is how, if we shut down specific genes in this environment, if we can influence regenerative events. Right, so I think you can see that reasonably well. Okay. Should we put down even these lights, I wonder? Where did my colleague go? Maybe you can see. Is there a light supposed to be there? These guys? Oh, okay. Is that better? Good. Okay. Yeah. I'll go to sleep, I guess. I don't think you will. So this is what it looks like, and I think that's instructive just to see what the proximal and stump actually looks like in reality. So these are neurofilament stains, and you can barely see them sort of progressing down this part. This is 7 days after injury. These are the Schwann cell profiles labeled GFAP. So not huge numbers, and the numbers have diminished dramatically as we get further down. So there is this idea that the growth is very restrained and reluctant. It's not as if you cut a nerve and 100 axons go flooding out. It's very much different than that. We think there are what are called pioneer axons as occurs in development. So a single axon may grow, sample its environment, and then others might follow along afterwards. So here again is what I've been talking about, transection, proximal stump, end bulbs. And this is kind of an ideal situation, but in reality, again, very restrained growth. Here are some axons and Schwann cells with them, very close overlapping. So this important relationship between the axon and Schwann cell. And then if you look through here, this is about 2 or 3 weeks later, under the EM, it's pretty interesting because you'll recognize here are some larger caliber myelinated fibers. But look at these clusters here, large groups of unmyelinated fibers. We think these may be sprouts from single parent axons. But why is this one already larger and myelinating? Presumably they started from the same kind of axon, whereas these are small. And this is an interesting process. Again, Tom Brushart first described in motor axons with Tessa Gordon, so-called staggered regeneration. So larger axons out earlier, they myelinate, and then other small ones come later, and these may be multiple sprouts from single axons. If there are any students in the audience, I think the best analogy to this kind of dispersion of axons is our division of neurology versus medical students. If you had a road race and you're standing at the end after an hour or so, all the medical students are so fit, and they'd come in as a big clump, racing each other all together. Whereas if it's the division of neurology and faculty, somebody may come in early, the junior faculty, and other people kind of stagger in late, and so you're waiting around for a couple of hours before the last people come in. So kind of a staggered approach to regrowth or dispersed growth. If you poison the Schwann cells, and we did this with mitomycin, Susan Hall had done this in freeze-dried nerve grass as well, and prevent them from proliferating or growing, both the axon and the Schwann cell growth is very poor and often misdirected. So whatever happens to one cell population, the axons, versus what happens to the Schwann cell, seems to go together. Early on, you cannot really rescue this if you add more basement membrane, which is what laminin is here. But later on, it may help a little. And this is just some of the data here. If you just focus on the black, and these are the mitomycin poisoned outgrowth zones from those proximal stumps, very much less outgrowth here. There is one fiber that made it through here somehow, which is interesting, always an exception, maybe a pioneer axon. Or if you look at the axons with Schwann cells, same story. If you just look at axons that have no Schwann cells, the number of fields, these are the number of fields from the proximal stump. So here's proximal stump growing further out. The number of axons dramatically declines. So axons don't like to do very much by themselves. They rely on their partners to be there and help them grow. And we'll get into some of the reasons for that. If you look a little later, here is a normal here's a normal environment of those bridges between proximal distal stump. Here's the small myelinated fibers that have been regrowing. This is the one that was poisoned with mitomycin. So there may be some unmyelinated fibers here, but very poor outgrowth. And we did rescue some a little bit with mitomycin. Okay, so I'm just going to highlight now some of the interesting molecules that connect axons and Schwann cells. This is a small sampling, and the numbers are growing, but they do offer interesting targeted opportunities to do things. And I think one important aspect of this is that some of the molecules have multiple roles. Very interesting. And for example, Richard and I were talking about VEGF yesterday, a vascular endothelial growth factor, but it signals both axons and vessels. CGRP we were quite interested in because we thought initially that if we crushed or damaged nerves, they'd be ischemic, and kept finding rises in blood flow in these nerve trunks, which puzzled us. And we found that that was partly because there was a release of the CGRP peptide in the environment of the nerve injury. And it's a very potent vasodilator, and it would vasodilate local vessels during repair. We weren't sure how essential that was. CGRP is expressed in axons as well. So it's the alternative splice product of the calcitonin gene, and it's almost exclusively in neurons. So if you crush or injure a nerve, as in BC through H, the levels of CGRP seem to rise locally in these axons. It's a normal nerve trunk and stain for CGRP, so very intense local staining. So we thought, well, that's interesting. So the CGRP must be leaving the axons, impacting on local microvessels, dilating. But, you know, what else might it do? So these are just some numbers of fibers, and I think near the edge of the injury and further out, the new fibers particularly express a lot of CGRP de novo. And then we did some work with Jeff Twiss, who was at the University of Delaware at the time, doing a combined immuno and in situ hybridization for message for CGRP. So the canonical idea is that protein peptide is produced in a cell body, and then it's exoplasmically transported out to the distal terminals, which could be a hassle sometimes because it might take a while for that protein or peptide to get there. But Jeff Twiss and Diana Willis and others, Christine Holt, have shown that in fact there is local translation at the very tips of axon. Very exciting and interesting phenomenon. And they showed that the mRNA is out here in the distal ends of regrowing axons here. So green is CGRP, or the red is the CGRP mRNA, and green is neurofilament. And this was just to show that they had nice pure axon preps here because it expressed beta actin, but not gamma actin. So are there other roles besides its effect on blood vessels? Well, Chanadol had shown that in neonates, CGRP is a Schwann cell mitogen, makes the Schwann cells proliferate. And so we reproduced this work in adult nerves as well and showed that CGRP acts as an axon to Schwann cell mitogen, increased Schwann cell proliferation. So interesting signal from the axon to the Schwann cell. Just one of a number of others. Neuregulin, you've probably heard of a little more, is in this category. And so what if you use those nerve bridges and you decide to shut down the mRNA for CGRP? Well, the result was very poor outgrowth. Here's our normal bridge growing. Not dramatic growth to begin with, as I mentioned. It's constrained and restricted growth. But if you put CGRP, siRNA in this environment and reduce the CGRP message, the axon outgrowth is very, very much worse and very restrained. Or you could do the same with the receptor. Okay, so just one example of an axon to Schwann cell signal in this environment. This is some interesting work where we took the idea that neurodevelopment is like a precursor of regeneration. So, and that's been a theme out there, and I think it's an interesting theme that you recapitulate developmental events during a regenerative process. So we decided to look at this signaling pathway with netrin. It's critically important in development. It operates on two receptors, DCC and UNC5H. This facilitates regrowth, and this is inhibitory to growth. But it operates at a neuronal level. So during development, there may be a point source of netrin. The axons grow towards it, and the receptors are on the neurons here. And here, if there's UNC5H, they're turned away. And this has to do with midline guidance and some really beautiful developmental literature that a number of you may know about. What about in the peripheral nervous system? Are the receptors present? This is work with Christine Weber, who is a postdoc in the lab, now up at the University of Alberta. We were surprised to see that the receptors, instead of being on the axons, were on the Schwann cells instead. So one important difference from neurodevelopment. What about UNC5H? Also separately expressed from neurofilament in axons on Schwann cells. And so she looked at Schwann cell preps and showed again that the DCC is present on the Schwann cell. So already a very different scenario than development. So here, and the Schwann cells play a much less important role during development. Here, the same machinery is being employed, but this time it's on a different cellular population on the Schwann cell. So what happens if you use an siRNA to knock down DCC? You get much poorer outgrowth here. Same model as I showed you before. And what happens to the Schwann cell happens to the axons. They do things together and in parallel. So once as I see the DCC present, the DCC is not present. I don't see the attractive cue. I'm not going to advance. This is a Schwann cell, and the axon agrees and does the same thing. What about, more interesting though, is UNC5H. So if you inhibit an inhibitor, might you have increased growth? And that's indeed what we saw with inhibition of UNC5H. So, and that has sort of become a theme as we move forward, as to finding out what are the breaks and holds up on this regenerative growth and going after them. So interesting recapitulation of development, but in a different way. The same machinery doing something similar, but this time the Schwann cell was taken on the role. A little bit about trajectory. I've mentioned this already before. Sorry if I'm talking away. I should move over a bit. During development and growth, the patterning and trajectory of axons has to be very precise. Otherwise, you're not going to form a nervous system. I mean, if you need to form a heart or something else, fine, but the nervous system, we need those exquisite connections so that you will remember, for example, this lecture at 2 or 3 days from now, a month. I wouldn't worry about it, but 2 or 3 days. So, and that's probably happening in adulthood as well, as you're remodeling synapses and maybe remodeling axons as well. We don't know a lot about it. But during regrowth, some trajectory is important. So it doesn't have to be the exquisite tight patterning and the laying down of trajectory cues and precise gradients. But we think it's still important because a misdirected axon is as effective as no axon at all during regeneration. So if all of the axons are misdirected, there's also going to be a poor regenerative outcome. And so the surgeons in the audience, of course, will know about these, which are interesting and kind of make the point, because these are what are called neuromas in continuity, proximal stump, distal stump. They're connected still. But if we did a section through here, there might be 0 axons. Here there might be 10, here there might be 100. So even though they're physically connected, the axons are not regrowing to the distal stump. And these are called neuromas in continuity, and then the surgeons need to decide, do we cut this all away and resew it? Do we put a sural nerve graft or perhaps one of these biodegradable tubes between these two? So why is that, though? If there is a connection, why do these guys not grow through this environment to the distal stump? They may circle around the epineurium. Here's some CGRP fibers that are circling around instead of going where they belong. After World War II, there was an interesting monograph published by Lyons and Woodall. So these were neuropathologists, and the surgeons were trying to cut out bits of nerves to try and rejoin them and to relieve neuropathic pain from neuromas. And these are some of the specimens they looked at. You kind of get an idea of the environment we're faced with. These are the fascicles that have become atrophic. There is connective tissue here. There may be ischemia from microvascular thrombosis. So very hostile, you get a very hostile impression of what's going on here. So here's a neuroma in continuity. The axons here course through nicely, and these guys just kind of stop. And then we have this whole environment. Is this secondary or is this primary? So that's the challenge of trying to learn how to overcome this. And I think in the spinal cord, people talk a lot about the connective tissue or the scar tissue. In the nervous system, in the peripheral nervous system, we're not sure if this is secondary to bad growth or is a primary inhibitor or whether it's ischemic. In some instances, it is. In our models, we can look at wrong-way axons, map out trajectory. So here's that experiment I showed you earlier with mitomycin. So you get increased numbers of wrong-way axons. If you give more basement membrane constituent, like laminin, you can reduce the number of wrong-way axons. So this is just a rough schematic of what may be happening here. So here is our growth cone, which rarely look like this in vivo, by the way. Some cues tell it to collapse on one side. Other cues tell it to regrow, perhaps towards the Schwann cell, which is then laying down growth factors. It's laying down basement membrane like laminin and fibronectin, and it's telling the growth cone to turn towards it and follow hither. Within the growth cone, we think that there are a repertoire of molecules that help determine its response. And so some of the earlier molecules looked at are ROA and RAC1. And again, these have become quite interesting from the spinal cord injury literature. So ROA is an inhibitor of growth, so it shuts down the growth cone. And we see it might be expressed on this side. RAC1, on the other hand, may enhance lamellopodia formation. And there's others that I'll talk about. TRACs are growth factor receptors that are involved in this. And P75 is a partner of a growth factor receptor. It was the original nerve growth factor receptor. We know partners with other proteins. So here's the idea that neurons are supported by growth factors, and these classically have been described from target tissue. So Rita Levi-Montalcini got a Nobel Prize for discovering nerve growth factor, and she and Stanley Cohen isolated it from most submaxillary glands. So NGF and others are taken up in axons, retrogradely transport, and support certain classes of neurons if they express the right receptor. We think the Schwann cell is in that category, that expresses BDNF and a whole host of other growth factors. The neurons themselves can synthesize growth factors, so-called autocrine support, and BDNF is a good example of that. And just quickly summarizing this family, NGF being the prototype, BDNF, NT4, 5, NT3, been known about for several decades now. But depending on which receptor is expressed will determine the response of the neuron. And this is kind of interesting. This is just two adult sensory neurons in culture, and we notice that their branches all seem to be diving in together to one common non-staining part of the graph here. And this turns out to be a cluster of satellite cells that were co-cultured with these neurons. So an interesting, attractive cue. We have started to try and address directionality in neurons, adult neurons. There is an assay that's been around in development for a number of years, invented by Yu-Ming Pu, called a growth cone turning assay, in which you have a growth cone and you can apply a picospritz source of growth factor support to it, and then you measure the directionality of growth. And so Christine, when she was in the lab, decided that we would set this up in adult neurons. Let me get this to work. There we go. And what she did is apply a picospritz source of NGF here. And these neurons were selected by an immunopanning method to be those that express the receptor for nerve growth factor TRAC-A. So these were TRAC-A, and we thought they might be fairly plastic. People think that small sensory neurons are a little more plastic than other populations. And so these are just kind of neat videos to show that over an hour, these are taken over an hour, that the growth cones do turn towards a picospritz source of NGF. But they have to be pre-injured, so they have to be what are called preconditioned or ramped up to do this. They have to grow on laminin. And we've done a lot of subsequent work on other populations, particularly what are called the GD&F responsive neuron population. And they're much less responsive to impacts like this. So there is some directionality, but it's not dramatic. And that's Christine. So we do think that trajectory is important during regrowth, as is successful regrowth. And then I'll talk a little bit more in the next few minutes about the intrinsic roadblocks to regenerative success. So I've already mentioned row A. So row A has to be connected. It has to be activated by GTP to be an effector, and what it does is acts on myosin and other constituents to shut down growth cones and restrict them from growing. So it's a member of the RAS GTP family, activated by GEFs and deactivated by GAPs. And just to highlight the pathway here a little bit, it operates through kinase, rho kinase here, but the bottom line is that it reduces axon growth through a number of players here. And there's family members, interestingly, which have completely different actions. So Rho A has become a target for spinal cord regeneration. Chu Cheng, my long-term colleague in the lab, did these experiments and first showed that Rho A is indeed expressed on sensory neurons here, particularly in the cell body, but also at the tip, where it might be particularly important. These are just dorsal root ganglion transverse sections, and you can see Rho A expressed in a number of the neurons here, stained with neurofilament. Also at these in vivo growth cones, so we see the Rho A where it matters, out in the periphery. It's kinase, ROC, or Rho A kinase, also expressed in sensory neurons here, and in dorsal root ganglia here. So both bits of the machinery present. And then if you transect a nerve, just walk you through this because this was really important, and this was Min Dang, a colleague down the hall from us, helped with this difficult kind of assay to measure activated bound Rho A. So if you cut a nerve, there's none distal. So we think the Rho A is mainly in neurons because there's lots of Schwann cells in this distal stump, but no GTP-bound Rho A. And so there's an upregulation of activated Rho A, so an upregulation of a brake. It's kind of counterintuitive. So you want regeneration to occur, but you've activated Rho A, which is going to restrain regeneration. Why should that be? Well, Rho A gets activated by a number of constituents in the CNS by myelin, so-called NOGO66 component of myelin, myelin-associated glycoprotein, semaphorins, and others. In the PNS, some of those constituents are important, but one item that comes up routinely and in large amounts is chondroitin sulfate proteoglycans, and we think these in the environment are one of the factors that turn on Rho A. If you put an inhibitor of Rho A, HA1077, and look from the proximal stump distally at a distance, here's our normal miserable outgrowth of axons here. But if you add the inhibitor at the same distance from the proximal stump, you get better outgrowth. And this is just the data here. And this is just a stain showing neurocan, which is a chondroitin sulfate proteoglycan, which is present in large amounts in this environment. David Muir and Greg Krakowski and others have shown that if you add matrix metalloproteinases that chew up CSPGs, you can improve outgrowth. So we think that's an important mechanism. So why would the nervous system put up a regenerative roadblock? Well, we think it goes back to development. And so it's this whole idea that you want growth to occur, but you just want it, you kind of want to control it. You want to restrain it and control. I guess it'd be like running a department of neurology. You want growth to occur, but you want just kind of the right people who are going to do the right things, and you want controlled growth. And you don't want inappropriate sprouting. So the next break we looked at is PTEN, and this was work with Kim Christie, who was a PhD student in the lab that David recalls, and then was a postdoc with us for a few years after that. So PTEN has become quite interesting. So this is a tumor suppressor molecule, phosphatase and tensin homolog deleted on chromosome 10. It inhibits cell motility, so that's important in cancer. Induces apoptosis, hydrolyzes PIP3, can be inactivated, and that's Kim. So this is a common downstream pathway. So if you remember, I was telling you about the cells, the neurons require the specific growth factor receptors. But if you wanted to kind of go downstream from that and manipulate the neuron itself with a very common pathway in many of the neuron cell types, one could go to PTEN. So it's shutting down this growth factor pathway to activated AKT right here. So we thought perhaps that if we inhibited PTEN, maybe we could also enhance plasticity of neurons. And so Kim was about 2 years into this project when, of course, Park and he published their science paper on PTEN knockdown, showing that in retinal ganglion cells in the CNS, if you knock down PTEN, you could enhance growth. So by the time we got to submit her paper, people were saying, oh, we know about that already. But what Kim had done is shown that it certainly is present in the peripheral nervous system. And some different twists to the story as well. So here are sensory neurons expressing PTEN in axons as well. You notice in the dorsal root ganglia, the expression is a little bit unusual. We think it's expressed in most of the neurons, but it's very highly expressed in the small population here. And I mentioned that the NGF responsive neuron population is quite plastic and grows a lot. Here's a regenerative break, and it turned out that this was expressed on the so-called non-peptidergic neurons, or IB4 population, which turn out to be slower growing, which is an interesting correlation. We think that may be important. So if you inhibit PTEN pharmacologically, as Kim did, you can increase outgrowth in adult sensory neurons in vivo. The interesting part of this was that there's a process called preconditioning. So if you injure a nerve and then grow it out in culture, the outgrowth is dramatically better than if you'd never touched it before. This is so-called preconditioning. Obviously not a great strategy for humans because you can't go around pre-injuring everyone's nerve, hoping that it will help if they have an accident later. But people are trying to mimic preconditioning, and cyclic AMP may have a role, electrical stimulation. But the interesting thing about PTEN inhibition is it worked better if they were already preconditioned. So it ramped up way beyond the preconditioning effect. So she repeated the experiments with an siRNA and had the same effect in vivo. And then we looked at these nerve bridges. So these are proximal stumps here, and this one was locally exposed to BPV-VIC, which is a pharmacological inhibitor of PTEN, increased growth, or with the siRNA as well. And then verifying knockdown, of course. Whenever you do siRNA experiments, you need to show that it's actually doing something. And this is the data. So like UNC5H, inhibiting this inhibitor enhanced growth. So this is our latest story on this whole chapter, because we've been pushing along the idea that there are really important commonalities between the unrestrained growth in tumor cells, which is what you want to inhibit and prevent, but the fact that neurons have the same proteins in them, but it's having the opposite effect, that it's restraining their growth and preventing their growth. And maybe if we can manipulate that system for short periods of time. We don't want oncogenesis to occur. Short periods of time in selected populations, we might be able to do something for regeneration. So this is work that's actually coming out on Monday. Retinoblastoma gene, long in the tooth. This work, again, Kim worked on it, as well as Dan Krishnan, another postdoc in the lab. So RB is a tumor suppressor protein. It operates by activating these E2S that induce transcription as well. In the retina causing tumors, a deletion or mutation of retinoblastoma in immature cells is responsible for the tumor. It is expressed in peripheral neurons, in the cytoplasm, but also in the nuclei here. It's not so much present in the Schwann cells. probably a little bit. Like PTEN, if you knock down RB, you can see increased outgrowth. We don't have pharmacological inhibitors for this, so the siRNA we rely on increases outgrowth, branches, process length. Bhagat Singh, also a postdoc in the lab, also repeated this in another species. So this is mouse as opposed to rat, which I just showed you, also increases outgrowth. And this is just the data. And then, I don't know if you can see that so well, but, and then used a completely separate system where we used floxed RB1 and then gave a adeno-associated virus that expressed CRE, which chops out these flox sites and allows the gene to be expressed, and showed increased outgrowth also with that method. So 3 different ways to show that it influenced adult neurons. And then, of course, to look at outgrowth as well. Again, locally in the environment, if you knock down RB1 with an siRNA, you can increase outgrowth. I'm not going to show you this, but we took it one step further as well and looked at further time course outcomes in a mouse if you knock down RB1. We have a brilliant undergraduate student, Kaylin Purdy, who did this and showed that if you knock down RB1, you can have increased maturation of fibers distal to the injury zone. You can have evidence of improved functional regeneration in terms of sensation, etc. Okay, well, I'm going to try and finish up quickly now by saying that I mentioned diabetic polyneuropathy, and that one of the goals for acquired neuropathies may be to improve regrowth, both from the neuropathy itself, but in diabetes there are challenges to regeneration. Diabetics have, say, carpal tunnel syndrome more commonly. We know regrowth, regeneration in diabetes is specifically impaired. So there are yet additional burdens. I've been making an argument that peripheral nerve regeneration is miserable and restrained to begin with, but if you add diabetes, which may be merging up to 10% of the population, the regrowth is yet more restrained. And then this final work is just to show that, going back to PTEN, diabetic neurons also respond to PTEN inhibition. And I'll skip over that a little bit, but just to say that diabetic neurons seem to upregulate this regenerative brake, this tumor suppressor. And this is work that we just have come out this year showing that if you knock down PTEN in a diabetic nerve and allow it to regenerate, the growth is improved functionally. So interesting new pathway for regenerative failure in diabetes. We've had a lot of ideas why diabetic nerves don't regenerate so well, everything from oxidative stress to microangiopathy. This is another pathway that may explain it. I'll skip over that. And I'll just finish off by talking about this process, which is called collateral sprouting. So when we think of regeneration, we're thinking of an injured nerve regrowing, but there are other mechanisms of regrowth, and collateral sprouting is an interesting one here that deserves some attention. So if you lose a neuron completely here, you can either try and put stem cells to regrow, or you can coax the remaining neurons to regrow and reinnervate adjacent territories. This is so-called collateral sprouting. Jack Diamond looked at this and thought that there were unique molecular requirements for collateral sprouting. These nerves in the skin are very plastic. Makes sense. You're shedding keratinocytes every day, so the nerve supply in the epidermis has to keep up and keep regrowing. And we see these interesting trajectories in the skin here of regrowing neurons. And so manipulation of this population would be particularly interesting. These are nerve cells around hair follicles. Just by cutting them, you can increase the nerve supply. And that probably involves the role of another growth factor called HGF. And then finally, we've been interested in the fact that insulin receptors are expressed on these small sensory neurons. And if you locally apply insulin, you can manipulate the outgrowth of these epidermal fibers. So just a taste of some of the other ways you can begin to think. So you can manipulate the cell body, you can shut down the tumor suppressors, but you can also do things locally to help our regrowth to occur. So what we'd like to know is whether some of the other players like PTEN, RB1, how effective they might be in just a local manipulative strategy Right. So Weir Mitchell was a peripheral nerve surgeon about 150 years ago in the American Civil War. No cases which we have been called to seem to us as sadly hopeless as injuries of nerves, but none has been rewarded by more enduring and steady efforts to afford relief. And that also goes for the neurobiology and the science behind this as well. And then my hero, Richard Feynman, says that, you just really have to think differently about this, and you have to come up with new ideas that are a little bit out of the box and may seem impossible. So I've had great input from a number of people. This is Jose Martinez, Wifeng Gua, Bhagat Singh and his wife, who've been part of this, and the whole team lined up in a place that David will recognize last year. And who's worked on the Row A story and on some new work as well. So I want to thank you very much, and I think we still have a few minutes for questions or whatever.
Thank you. Both Nature and Science have had recent commentaries about problems with mouse and other rodent work that are very promising, lead to all kinds of drugs that don't work in humans. And I'm not implying it, mouse and rodent work shouldn't be done, but that there are so many things to do with humans. So, for example, with DTI, we can see in this work, some of this work has been done here in radiology. You can see the nerves, and I imagine you could see if they're regenerating with DTI, and there are maybe 8 million papers about brain metabolism, brain blood flow, brain neurochemistry with PET and MRI. But what about metabolic or blood flow studies in nerves?
Okay, yeah, so several
human nerves.
Yeah, several components to that. I think, I think the discussion is timely and important, and I agree with it. I don't think it should be an argument to shoot down the critical importance of preclinical work, because there's been really some spectacular breakthroughs with the rodent models and mice and rats, but that's a huge debate. In diabetic neuropathy, it's particularly relevant, though, because a lot of studies have used these short-term models of 1 month, and then they get the latest treatment that reverses diabetic neuropathy. But, 1 month in an animal versus a human, it's not a great story. I think the imaging, the second part of that is about the imaging studies, and I think they will come along. I have yet to see an image of peripheral nerve axons with DTI, individual axons to see what's going on. I think it'll come. I think it's appropriate. There are some really nice examples of the translation, though, from preclinical to human with electrical stimulation, these brief epochs. I didn't talk about this for 20 minutes. If you stimulate a proximal stump, you have enhanced regrowth. Work from, again, Tom Brushard, Tessa Gordon, Valerie Verge, and others, and we've worked on this as well. And Ming Chan and the University of Alberta has shown that in humans, the same paradigm has a very interesting effect and improves regrowth. And it may be because it's turning on those regeneration-associated genes very quickly and proximally, like a preconditioning effect. So I think there are some really important examples. Let's see, was there one more part of that, the DTI and...
PET or other...
metabolic and nerve blood flow studies. Yeah, so I started out my career doing physiological measures of nerve and ganglion blood flow. And the idea there was that people thought that nerve blood flow reductions caused diabetic neuropathy. And there's still big arguments at meetings about this. And there, in some models you can demonstrate it. We couldn't see it in other models. In the peripheral nerve, the blood flow is highly redundant. Lots of vessels. You can reduce flow in the nerve spine for a while, and the surgeons mobilize big, long chunks of nerve. There's no auto-regulation in the peripheral nerve. In the ganglion, though, there is some degree of auto-regulation. So it's almost as if the metabolic demand changes the relationship with blood flow. Does an ischemic nerve regenerate more poorly? Probably, but it has to be really very ischemic, I think, to do so. And do you need more blood flow for these regenerative events to occur? Not sure about that, how much more you need or what. And Richard and I were talking about this yesterday. I still don't know the answer to this, I think. But all very good points. Thank you.
So I have a question that we always care about the intrinsic growth factors which affects the nerve regeneration, right? So we look for all those factors. But as I was saying, when you stop the Schwann cells proliferation and stuff, the effect is much more prominent. So what about these intrinsic growth factors which affects the proliferation of these Schwann cells out there? And I think that that's what is also helping when you are doing some kind of graft in the place and helping in the regeneration. So what's your thought about that? That, these blocks could be also in the Schwann cells, which is affecting the regeneration. So what's the take about that?
Yeah, no, I think it's a very good point. And we were, the argument was that the relationship is very, very close. So there are things you can do to make the Schwann cells grow better, CGRP being one, and neuregulin being another. But people are also engineering Schwann cells to put them in these grafts. And my colleague Raj Mehta has been working on skin-derived Schwann cells, so they can reprogram skin cells into Schwann cells and adding those on as immature Schwann cells. They seem like they're fairly potent factories of growth factors, so they do a lot of interesting things. So adding a pile of Schwann cells is probably a good idea in some instances. Now, keeping in mind that with a lot of neuropathies, it's the axon or wiring that's disappeared, and there's plenty of Schwann cells still around. They do proliferate and they're there, and if you cut the nerve, the Schwann cells begin migrating into the cut zone from the distal end and the proximal end, so they do flood there. So do you need that many more? Not sure. I think his work does look promising, though. So you're right.
So one of the consequences of peripheral nerve injury is that the sensory neurons become sensitized to the pain signals. Do you enhance growth? Does that affect physiology of the sensory neurons at all?
Yeah, I think there's a relationship that I don't understand completely. So when the nerves don't grow and they form these end bulbs, Marshall Devor and others showed that sodium channels accumulate on those little end bulbs, and we think that they generate ectopic activity and produce pain. And he could shut that down with things like dilantin or lidocaine. So I think axons that have been frustrated and don't grow change in ways that facilitate pain. They also express opioid channels, which is interesting. So you can use local opioids just in this environment without any CNS side effects and influence some aspects of pain. Bradykinin probably plays a role there as well as other factors. We do think that more successful regrowth with time dampens neuropathic pain, and I think we've seen this in the so-called CCI neuropathic pain model. This is a model where you put these loose ligatures around a nerve, and over 3 or 4 days, the animal becomes hypersensitive. It's Bennett and Z identified this, and it's really been an important model of neuropathic pain. But it only lasts for so long because the axons eventually grow out around the sutures and reinnervate, and the pain behavior goes away. So there are these really interesting and important relationships, but there's probably a lot more. I think,
So during regeneration and collateral sprouting, the thermosensitive and pain-sensitive C fibers have a relatively easier time growth. What about the A fibers, particularly those that are non-nociceptive? Are they able to regrow out into the peripheral target tissues, and also are they able to regenerate their specialized nerve endings, their Pacinian corpuscle, or Ruffini endings?
I think it's a very good question, and we don't have all the answers. We do know that over time, some of the end organs disappear, like Pacinian corpuscles or Meissner's corpuscles, and so there's nothing for the axons to find. But it's possible new ones can be created and it can be reinnervated. So I guess the answer is some occurs. It's not ideal. And I don't think the patterns, there's been some work on the patterns of regrowth, but not a lot of work on this. So it is an interesting area. Epidermis, a little bit easier. You can see all the populations. And innervation of the hair follicles and sweat glands, also very interesting. I think there was some work I've seen that I can't remember if it's Pacinian corpuscles where the axons get back, but they don't quite form the same kind of connections with the end organ that they did before. So, and I think there's also been descriptions of people with nerve repairs that the sensation never seems quite right in terms of how the growth occurs. I'm always puzzled by ulnar neuropathies, where you have this, classical loss of sensation and splits half the finger, and these last forever. And, why wouldn't the axons from the median nerve just regrow this tiny little distance over to the other side to reinnervate the skin? I don't know. Interesting, though. It's a good question. Thank you.
Okay, thanks. That's